Hello, dear learners. So, the next topic of our discussion is ELISA. ELISA means enzyme linked immunosorbate assay, which is the widely used serological technique. It is used to detect and quantify the presence of antibodies and antigens in the samples. ELISA is commonly used in clinical diagnostics, research, and food safety testing. एलिसा टेक्निक को हम यूज़ करते हैं एंटीबॉडीज़ और एंटीजेंस के डिटेक्शन के लिए सैंपल में. Types of ELISA. There are majorly four types of ELISA, that is direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. So what is direct ELISA? Direct ELISA detects antigens directly without the need for secondary antibodies. Indirect ELISA detects antibodies by using a secondary antibody that binds to the primary antibody. Sandwich ELISA, it detects the antigens by using two antibodies that sandwich the antigen in between them. What are the steps of typical ELISA? The, tip, the typical ELISA steps include coating, blocking, and incubation. Coating. The antigen or antibody is coated onto the surface such as microplate will. Blocking. Non-specific binding sites on the surface are blocked to prevent false positives. Incubation. The samples containing the antigen or antibody is added and allowed to bind. Washing. Unbound molecules are washed away to reduce background noise. Detection. A secondary antibody conjugated to an enzyme is added to detect the bound antigen or antibody. Colorimetric reaction. A substrate is added that reacts with the enzyme to produce a color change. What are the advantages of ELISA? The advantages of ELISA are that it is the high sensitivity and specific. It has the ability to detect multiple samples simultaneously, and it can obtain quantitative results. ELISA tests are generally relatively accurate tests. They are highly sensitive and specific. They are used in wide variety of tests. Antigens of very low or unknown concentration can be detected since capture antibody only grabs specific antigen. It is generally safe and do not require radioactive substances and contains diluted sulfuric acid. What are the limitations or disadvantages of ELISA? Limitations of ELISA. The false positives or false negatives can occur. Cross-reactivity can lead to false results. It requires skilled personnel and specialized equipment. Measurement of enzyme activity can be more complex than measurement of activity of some type of radioisotopes. Enzyme activity may be affected by plasma constituents. Kits are commercially available but not cheap. Very specific to a particular antigen won't recognize any other antigen. False positives, negatives possible especially with mutated or alterated antigen. ELISA in clinical diagnostics. ELISA is used to detect infectious diseases such as HIV and hepatitis. It can also be used to monitor autoimmune diseases such as lupus and rheumatoid arthritis. ELISA is a key tool in diagnosing and controlling disease outbreaks. ELISA in research. ELISA is used to study protein-protein interactions. It can also be used to measure cytokine levels and other biomarkers. ELISA is a valuable tool for drug discovery and development. ELISA in food safety testing. ELISA is used to detect allergens in food products. It can also be used to detect pathogens such as E. coli and Salmonella. ELISA is a critical tool for ensuring the safety and quality of our food supply. ELISA and COVID 19. ELISA is being used to detect antibodies against SARS CoV 2 in patient serum. ELISA can also be used to measure levels of cytokines associated with COVID 19. ELISA is an important tool in understanding the immune response to COVID 19. Future of ELISA. Advancements in the technology are leading to an increased sensitivity and specificity. New developments such as microfluidics and digital ELISA are on the horizon. ELISA will continue to play a vital role in diagnostics, research, and food safety. Conclusion. ELISA is a powerful serological technique which is used to detect and quantify antigens and antibodies. It has a wide range of applications in clinical diagnostics, research, and food safety. With ongoing advancements, ELISA will continue to be an important tool in the years to come. Hope this lecture was useful. Thank you. Happy learning.